going to be talking about advanced operando spectroscopic techniques for electrocatalysts in the first half, and then Andreas is going to be looking at photoelectrocatalysts in the second half. So a brief outline is I'm going to go over some kind of introduction very briefly, and then cover 3 different kinds of spectroscopy, starting with X-ray, vibrational, and then for the rest of today, we'll be looking at electronic spectroscopy. And I'll go over some basic principles and then focus on mostly examples. And all of the examples, just to keep it simple, today will be based on the water oxidation reaction, which is the anodic reaction that occurs during water splitting. And it's really kinetically challenging, which then obviously means that there's a lot of efforts currently in trying to understand these different reaction mechanisms that occur during water oxidation. So just starting off with why do we bother with all of these different techniques? So the basic reason is that so far what we've seen is just electrochemical JV curves. So you measure the current as a function of potential, and there's all sorts of theories where you can interpret why the current changes one particular way as a function of potential. And that's great, but what it doesn't tell us is what exactly is happening at the electrochemical interface, which is really important if we want to try and develop better electrocatalysts and want to get a more active or more stable catalyst for these reactions. So in situ spectroscopy then allows us to get better insights into things like the active sites, the reaction intermediates, also things like reaction pathways, and also getting to understand a bit about the interfacial reaction environment. And then what we can also do is probe the catalyst structure and composition before and after the electrochemical, or even during the electrochemical reaction, to see how this changes as a function of potential. So starting off with the basics, what is spectroscopy? So in its kind of most traditional definition, spectroscopy is the study of interaction between matter and electromagnetic radiation, either as a function of wavelength or frequency. And if we were to just look at this definition, we can see that spectroscopy has been around for a really long time. So here on the top left now is a picture of Newton, where he's using sunlight and passing it through a prism and seeing the different colors of light. So this is kind of the one of the most first examples of the understanding of interaction between light and matter. By the 1800s, people had moved on from studying things with sunlight, and they were putting these different elements in a Bunsen burner and seeing that you can have different colors with different elements as you burn them. And people were using that to figure out what different elements were present in a particular material, and also then quantifying the amount of those elements in a material. Today, of course, you have much more sophisticated techniques. You can have tunable X-rays at a synchrotron. You have really good detectors, really good optics, and you can do a lot of in situ characterization and look at these really challenging interfaces, especially in this case for electrochemical reaction. But in essence, this kind of technology has been around for about 400 years, and we're just getting better and better at doing some of these different studies. So first, what I'm going to start with is X-ray spectroscopies. So X-rays are really quite energetic. The energy range, broadly speaking, is somewhere between hundreds of EVs to hundreds of kilo electron volts. And because now X-rays are quite energetic, what they can do is they can excite co-electrons in a given material. And then given, based on the different processes that you might be looking at, you can basically categorize these effects into different spectroscopies. So for example, in X-ray photoelectron spectroscopy, you can just take this core hole and excite it out of the material, and you basically measure this kinetic energy of this core electron as it's emitted from this material that you may be looking at. If you were to look at X-ray absorption spectroscopy, here you're basically taking this core electron and exciting it to an unoccupied state in the material. And then X-ray emission spectroscopy is looking at the relaxation of the system once it's been excited. So you're taking then an electron from the occupied state and putting it back into this core hole that you've created when you did the initial excitation. So I think Mary, in the next lecture or so, is going to go over X-ray absorption and X-ray emission in a lot more detail. So I'm just going to touch briefly on X-ray photoemission spectroscopy in the first part of this talk. So in X-ray photoemission spectroscopy, like I said before, you're measuring the number of electrons you have for a given kinetic energy. And then you get a spectra that looks something like this, where you're seeing the x-axis as a function of kinetic energy or binding energy, and measuring basically the number of electrons that you have emitted. And on a lab source scale, this is usually an ultra-high vacuum technique, but one can go to a synchrotron where they have more sophisticated kind of differential pumping systems, where you can still keep your analyzer at ultra-high vacuum, but pump relatively high pressures of gas, going up to 1 torr. In a couple of cases, people can even go up to a couple of torrs now, and then you can basically measure these spectra as a function of the partial pressure of these different gases here. So one example is work that I've been looking at in my PhD, which is the interaction of water with rutile ruthenium oxide. And this is, for example, an oxygen 1s spectra as you dose water in the chamber. So starting from 0.05 torr and going all the way up to 1 torr. And you see that as you dose water in the chamber, you have these different species that evolve with this with the increasing relative humidity. And in order to try and understand what these different spectral features mean, you might need to know a little bit about what the surface looks like. So it's quite a simple oxide. You have 2 different sites. You have this coordinatively unsaturated, or this Cuss site, and then you have this bridging site, which is basically filled with oxygen under stoichiometric conditions. And most interestingly, at least from an electrochemical perspective, is this case where you have really high water vapor pressure. So that mimics most closely this kind of electrochemical environment where you have an entire layer of liquid on the sample. And here what we see is that this intensity of this peak of the Cuss site is about twice that of the bridge site. And what that can do is it can help us develop this model of how water might interact with the surface, where we propose that you have this kind of half dissociated structure where all Cuss sites are filled with either water or OH, and then on the bridge site, you have only every second site filled with OH. And that then captures well this kind of 2-to-1 ratio between this peak intensity here and then the peak intensity being much lower on this bridge site. So while this is already quite useful in trying to understand how water might be interfacing with some of these materials that are relevant for water oxidation, what you want to try and get to is a system where you can actually have a liquid layer of water on the sample, and you actually applying electrochemical potential and then being able to see the speciation as a function of potential. So with that in mind, there have been some developments in that area, and this is just one example that we were looking at a couple of years ago. This is a perovskite, which is also a good water oxidation catalyst. And here with this kind of dip and pull technique, you can stabilize a really thin meniscus of electrolyte basically by surface tension. You dip this sample in the electrolyte, and then as you pull it out, you have this really thin layer of electrolyte. And then you can basically apply potential and do measurements that way. But when you start seeing the spectra, one can then start appreciating how challenging this might be, because you have these huge peaks that come from water in the gas phase and water in the liquid phase, which can basically dwarf species like OH or carbonate, which also have oxo groups in them, and they might be really interesting from perspective of trying to understand the reaction mechanism, but their intensity is now significantly lower because the spectra is dominated by peaks from water in the gas and the liquid phase. But there's still a lot of developments in this area. People are trying to come up with different cell geometries and different ways of trying to do this measurement so that we can minimize the influence of these peaks and kind of focus in on what we might be really interested in. So that was a kind of brief overview of what you can do with X-ray photoelectron spectroscopy. The next thing I wanted to talk about was surface diffraction. So this, in kind of traditional definition, is not really a spectroscopy because there's no significant, or there's no change in energy between the incident and the reflected X-ray. But here you're basically relying on Bragg's law, where under certain, if you have certain conditions for this incident X-ray, you can reach a condition where you have perfect, all your reflected X-rays basically in phase, and you can have a much brighter intensity because of this constructive interference. If you meet those conditions, and those then depend basically on Bragg's law, which is a function of d and this angle of incidence here. And so most of you might already be familiar with bulk X-ray diffraction. This is just lab scale XRD of powder samples that probably some of you have looked at. And that basically relies on this approach where you assume that you have an infinite crystal, and then at discrete L values or theta values, as some of you might have seen, you have these regions of infinite intensity at these discrete L values. But what we have for a lot of electrochemical reactions is not this idea of an infinite crystal, but what you have is a truncated surface where you carry out these different electrochemical reactions. And now when you have a truncated surface, what you have is not just these areas of infinite intensity at discrete L values, but you also have this area of where you have this non-zero intensity between these L values. And this is basically called the anti-Bragg region. So where you have this kind of infinite intensity, that's the Bragg point, and then these regions in the middle are the anti-Bragg regions. And because they are formed by virtue of having a surface, they can then be sensitive to different things that happen on the surface. So for example, if you have surface reconstruction, or if you have different adsorbates forming as a function of potential, you can expect to see changes in the shape of this anti-Bragg feature here. So looking at an example again, how you can use this technique. So this is again a rutile ruthenium oxide single crystal surface with a really well-defined cyclic voltammogram. This is what one of these single crystals would look like, and you can just study one facet at a time. So here's just an example of what one of these L scans would look like, just comparing the low potential and the high potential just prior to the evolution of oxygen. And like I said, if you were to go to a synchrotron light source and you can really capture this low intensity anti-Bragg region, you can see that it has significant changes as a function of potential. And then what we can do is you can construct a model and basically move around these different atoms and use a genetic algorithm to fit this data. And you can basically get structural positions of these different atoms that match this experimental data the best. So we're, of course, in this case, interested in the water oxidation reaction. So we are really interested in the structure at 1.5 volts, just prior to the evolution of oxygen. And what we see in that case is that we have this OO group that we form on one of the active ruthenium sites. And these are the bond lengths that we can refine using experiment. And using theory, we can also see that this OO group is the most stable structure at these potentials, where it can basically be stabilized by this neighboring OH group, basically this configuration here. And what that then tells us is that the rate determining step for this reaction might be the final deprotonation of this last hydrogen bonded to this OO group. So if you were to increase the potential slightly, you can then deprotonate this hydrogen, and then that results in the evolution of oxygen on the system. So here we were basically looking at how different surface adsorbates can change as a function of potential. But what you can also look at is how different ions might order at the interface. So for example, here, if you were to look at either ruthenium dioxide or platinum, but now probe much more into the bulk of the electrolyte, you can see that as you polarize the surface negatively, you have a negative surface charge, and then you can basically adsorb or you can have ions such as cesium, which are relatively heavy, so they have a significantly large electron density, which can be captured by this technique. And you can see that you have this intensity growing at about 4 angstroms into the electrolyte, and that basically comes from the ordering of these cations at the interface. So this is a really powerful technique, especially if you have really well-defined single crystal electrodes, where you can then be able to not only detect surface intermediates as a function of potential, but you can also try and start getting insights on how ions might be ordering in the double layer and how that can affect reactivity. So that was a brief overview of X-ray techniques. The next thing we're going to be looking at is different vibrational spectroscopies. So specifically, for example, infrared radiation has really low energy comparing to what we had for X-rays. This is on the order of a few milli-EVs to a few EVs. And because of the low energy, what they can do is only excite between different vibrational energy states. So the simplest kind of infrared absorption spectroscopy is just exciting between these two different vibrational states. A related spectroscopy where we can also, for example, look at vibrational modes of molecules on the surface is Raman spectroscopy, where, for example, if you were looking at Stokes or anti-Stokes scattering, what you're doing is starting from a ground state, exciting to a virtual energy state, and then you have a relaxation, but the final state and the ground state are not completely the same. So you still have a kind of change in the vibrational energy state, which is what you can probe here. So it's slightly different from infrared absorption spectroscopy in the basic working principle, but it's quite similar in the kind of information that you can get using this technique. So here's what one of these cells would look like, where you have this kind of 3 electrodes set up here, and at the bottom you can have an IR beam come in and go out, and you can measure what happens on this electrode as a function of potential. So here's that example of ruthenium dioxide that I showed you before, where using surface diffraction, we were able to figure out that you have this OO group on the surface. Another technique you can do to prove the presence of this group is infrared spectroscopy, where we know using DFT that this has this classic kind of vibrational mode at about 900 wave numbers. And we see here's a representative spectra where we increase the potential, and then at 1.5 volts and beyond, we see this vibrational mode, which we can assign to this mode as we'd expect using surface diffraction, and that kind of confirms this mechanism that we were proposing for this material here. So another example is a homogeneous catalyst here, which is cobalt oxide, which is again a good water oxidation catalyst in this case. And here we have, it's a light-driven reaction, it's a homogeneous catalyst, but we can see here we have these ruthenium light sensitized complex, but they're not really important for what we are looking at here. Their only role is to kind of shuttle charges to and from this cobalt oxide center. And similar to what I showed before, you're able to, in this case, figure out that there's 2 different vibrational modes. You have this one appear at 840 wave numbers, which is assigned to this cobalt oxo vibration. And then you have this other mode here appear at about 1000 wave numbers, which is this complex OO intermediate on the surface. And then in order to figure out how these 2 different intermediates relate to the overall reaction mechanism, what the authors do here is a kind of pump probe spectroscopy, where you have the light on for a short period of time, and then you basically turn the light off. So looking first at this OXO, the time-dependent behavior of these OXO species here, we see that you have this pulse of 300 microseconds where you have the light on, and using mass spec, what these authors can see is that they already have the evolution of oxygen within this 300 millisecond time frame. And then as soon as you turn the light off, there's this really slow decay of this species. And what they attribute it to is this cobalt oxo then converting to this next step in the reaction cycle, which is this oxyhydroxide intermediate. But like we can see here, this is something that takes thousands of milliseconds, and since this process is so slow, it can't really explain why why they're able to see oxygen in this 300 millisecond time frame. So in order to do that, what they then propose is that you have the second cycle, which involves this OO intermediate with this vibration at about 1,000 wave numbers, where after you turn off the light pulse, you see that this intermediate is just accumulated on the surface and basically just stays there until you have another light pulse, which can then trigger this final deprotonation step to form evolved oxygen. And this again is quite similar to the mechanism that we'd seen a couple of slides earlier on ruthenium oxide, where you have the buildup of these OO species on the surface, and then you have this final deprotonation being rate limiting. So hopefully that gives you some insight into how powerful IR spectroscopy is, especially when you couple it with time-dependent measurements to give you some insight on the different species that might be, the vibration of these different species that might be present under reaction conditions. So for the next part of, I guess, today's lecture, we are going to be looking at UV vis spectroscopy. And these are basically based on either ultraviolet or visible radiation. So they're not as energetic as X-rays, which is on the order of several hundreds in kilo electron volts, and they're not as low energy as infrared rays. So what they're able to do is, at least for the systems that we're interested in here, is basically cause DD transition. So you're looking at electronic transitions using the spectroscopy. So this relies on some knowledge of crystal field splitting. So for example, if you had a transition metal ion, you'd have degenerate d orbitals. And then in the presence of a ligand, you can, so in this case, you have, for example, an octahedral ligand field, and you can split those degenerate d orbitals into, for example, T2G orbitals and EG orbitals. And then you have the magnitude of this delta, which basically governs these different DD transitions, can depend on things like the oxidation state of the metal ion, the nature of this metal ion, and then, of course, the number and geometry of these ligands, as well as the nature of these ligands that you might have for your specific complex. And this is quite an interesting spectroscopy if you're particularly to look at some of these oxides, which also happen to be a lot of the oxides of interest for different electrochemical reactions. And they exhibit this either cathodic or anodic coloration, which we can use to our advantage to study how their properties change as a function of potential. And this is something that's already been used, for example, more technologically in electrochromic windows, where you just apply electrochemical or electric pulse, and then you can see because of the electrochromism effect, you have a change in the color of the glass here. But in our case, of course, we're more interested in studying the electrochemical response of these oxides. So we are applying electrochemical potential, expecting a current response. But we also have, since we have this electrochromism effect, we can use that to our advantage to understand something about the reaction mechanism. So here's a basic setup of what, and on the top left, here's something that it looks like in the lab. Here's a simplified schematic of what it looks like. Again, it's basically a working 3 electrode setup, and then you have a light source. You have a lot of different optics here, which have not shown for simplicity, but you have the light source, which can transmit through the sample, and then you detect the emitted light, so the light from the sample, basically. And what you can do is at the same time, you can measure the electrochemical response. So you can apply a potential and measure the current as a function of potential, and at the same time measure the absorption spectra as a function of wavelength. And putting both of those together, you can then hopefully start to correlate electrochemistry with changes in absorption. So there's 2 distinct things you can do. You can either look at how spectra changes and how spectral features change, or you can look at the kinetics of these processes, again, like the infrared example, do time-dependent spectroscopy. So I'm going to look at both those separately. So starting with how we can just interpret changes in spectral features. And that's what we're going to look at first. So for example, this is water oxidation on nickel iron oxyhydroxide. And this is, you have iron oxyhydroxide and then iron incorporated in a nickel oxyhydroxide matrix into either small levels of doping or significantly large levels. And here you can see by measuring the spectra as a function of potential, there's two distinct things that one can draw from this kind of analysis. The first is that the spectra at low potentials looks quite distinct from that at high potentials. So there's probably two different oxidation processes, and one can then use this kind of spectroscopy to distinguish between these two different processes. Another interesting thing is if you were to look at the spectra at high potentials, you see that the iron oxyhydroxide spectra looks quite close to this this catalyst here, where you have a significant amount of iron doping, which suggests that the active center might be based on the iron site. However, when you have much smaller amounts of doping, you have a spectra that's quite drastically different, which suggests then here, in this case, you might have an active center that's centered around basically the nickel atom here. You can also track how these absorption changes as a function of potential. And you see here that as you increase potential within the water oxidation region, you have an increasing amount of this absorption. And Andreas in the second half will be talking about how one can correlate the kinetics to the density or to these changes in absorption that we have as a function of potential. So in another example, what you can also do is more than just trying to understand the active site, is try to understand the active phase of the material. So this is again an example of a cobalt-based catalyst. So it's a zinc-doped cobalt oxide, which starts off with the cobalt being in a tetrahedral coordination environment. So in the dry spectra, you see you have that in purple here. And as soon as you dip it in electrolyte without kind of any oxidation or reduction, you see you have a shift in the spectra to lower wavelengths or larger energies, basically. And what these authors have proposed is that you have a change in this tetrahedral coordination environment of the cobalt to the cobalt forming this kind of oxyhydroxide structures, which then has an octahedral coordination environment and then has this much larger splitting between these d orbitals. So you can see it moves to the left or to the lower wavelengths or higher energies because of this octahedral ligand field. And then again, you can see how there's changes as a function of potential. You have this electrochromic effect similar to the previous slide, where one can then quantify how the increase in absorption can affect the kinetics of the reaction. So the final example here I wanted to show was iridium oxide, which is something we've been working on currently. And this is a really challenging oxide because it has multiple redox states and it's really difficult to figure out the active site within this material. So there have been some DFT calculations about 4 or 5 years ago now, where what they were able to see is that if you had a single iridium center, you have quite a flat UV vis spectra, and one doesn't expect to see any significant absorption changes. But if you look at a dimeric center, so two neighboring iridium sites connected by these oxo molecules, you can then expect to see significant changes in the UV vis spectra as you oxidize this dimeric cluster. So in order to see what this looks like experimentally, we can synthesize, in this case, it was an anorically electrodeposited iridium oxide, and measure the absorption changes as a function of potential. And what we see is that we have these really distinct features that we can see in the absorption spectra, loosely proving this hypothesis that you might be having a dimeric or a multi-iridium center that's connected by oxo species that's responsible for the catalytic activity of the system. So if we were to look at the spectrum more closely, we can see that at low potentials, we have a growth in the feature at about 600 nanometers. And here kind of the pure component spectra corresponding to these changes. So the first transition has this feature at about 600 nanometers. And then going to more intermediate potentials, you have this increase at about 750 nanometers or so. And then finally, at water oxidation conditions, you have this distinct spectral shape with this feature growing at much lower wavelengths of about 500 nanometers. So based on these pure spectra, one can take these and using kind of a global analysis method, you can fit a linear combination of these three different spectra to the data that we have here. And what you can end up with is how the population of these different redox active intermediates changes as a function of potential. And then going back to this dimeric model that one can look at, we can then propose that you first have an oxidation of one of these iridium centers during this first redox wave, and then you have an oxidation of this second iridium center, and then finally you have a third oxidation, which results in this active state for water oxidation. So these three examples we're just looking at how one can just look at the spectra, visually see changes, and use that to interpret something about what might be happening in terms of the mechanism for water oxidation. What you can also do is try and look at time-dependent spectroscopy, and that can help you both quantify these changes and look at kinetics of some of these key relevant processes. So for example, if you wanted to quantify what these changes in absorption corresponded to, you could do pulsed voltammetry and couple that to transient absorption spectroscopy. So in pulsed voltammetry, what you'd be doing is applying a potential V and then stepping up a potential V1 and then coming back to a potential V. And this is what the current time response would look like. When you step down, you see you have this reductive charge, which corresponds to the reduction of these species that you're generating in your transient absorption spectra. So if you had a much larger potential jump, you'd form many more of these states on the surface, and that would correspond to a larger reduction charge that you have here. And so basically you can integrate the number of electrons transferred here, and then if you had some prior knowledge about the system, you could figure out how many electrons it takes to form one of these oxidized reduced species, and then correlate basically the absorption changes to an absolute change in the number or the density of these species on the surface. And that's really useful then if you wanted to try and look at turnover frequency. So in this case, it's defined as the number of oxygen molecules per site. So you can, like I said, you can convert all the absorption data to the number of these species that you have per second. So this is kind of an intrinsic method or an intrinsic activity metric. And here you see for all of these different samples, nickel oxyhydroxide samples, you have a similar dependence of the turnover frequency on potential. And if you were to just look at the electrochemistry, you wouldn't necessarily reach the same conclusion. So here you have quite different Tafel slopes going all the way from 30 to 50 millivolts per decade. And just based on this data, you might be tempted to then say there's possibly different mechanisms at play, but once we correct for how the density of these species changes as a function of potential, we see that they're all following quite a similar mechanism in this case. The other interesting thing you could do is what these authors have done here. So they stepped from 1 volt to 1.5 volts and then step down from back to 1 volt and track this species or track the kinetics of this species, which basically corresponds to the nickel oxidation, and see how that changes as you dope iron in the sample. So it's quite evident here that as you start doping iron, the time constant for this nickel oxidation becomes slower and slower. And what that tells you is that doping iron might to some extent benefit the intrinsic water oxidation kinetics, but it negatively impacts how quickly you can oxidize one of these nickel centers. So then that gives us some information on how, for example, the potential determining step of nickel oxidation might be changing as you dope iron in the system. So the final thing that I wanted to look at is a special case of these voltage step measurements, where here you start with a voltage V, you can step up to a voltage V1, And then instead of stepping back down to the initial voltage, you let the system relax back to open circuit potentials. So this is, in this final stage, you have zero current, and you just let these intermediates that form chemically react to basically turn over and form oxygen. So schematic in this case would be that initially you have a low voltage and you have a low coverage of oxidized species. As you increase the voltage, you have a much higher coverage, and then you can basically let the system go to open circuit. And then this initial rate, so in some cases, these oxo species might already be able to chemically combine to form evolved oxygen, for example. And then this initial rate of decay depends then on the density of these oxidized species and provides kind of a metric for water oxidation kinetics under these conditions. So one can then get that initial time constant and use that to get useful information about the kinetics of the reaction. So this was kind of alluded to in the late 1950s by the work of Conway on nickel oxyhydroxide electrodes, where here you can see as you leave the system to go to open circuit or decay to open circuit, you can detect that you have evolved oxygen where these oxo species might be chemically combining to form this evolved oxygen. So what you can do is, again, like I showed before, you can look at the kinetics of this decay and figure out what the time constant for this water oxidation might be. And here we see it's about somewhere between 0.5 and 1 second based on these open circuit decay measurements. And this corresponds quite well with what one might expect to be the turnover frequency for water oxidation on these materials. So then I think Andreas is going to talk about this in the second half. Then one can start looking at how water oxidation kinetics change with things like electrochemical potential, as well as the density of these species that you're able to accumulate before you let your system go back to open circuit conditions. So hopefully that's gives you a good overview of what you can do with UV-vis spectroscopy in terms of trying to understand these electrocatalysts, both from just a kind of steady state approach where you're looking at changes in the spectra, and then the approach where you're looking at time dependent changes and then looking at kinetics. But before I wanted to finish, I really wanted to highlight these two examples here, which really hopefully drive home the point that for all of these spectroscopies, you are perturbing the system. You have an incident source of energy on the system, and one really needs to be careful and have control experiments in place to kind of figure out or to kind of confirm that you're not having any influence of this incident radiation on the actual reaction that you're carrying out. So here's an example where you have a laser Raman spectrometer. But while you're doing your reaction, if you were to put an IR camera on your sample, you see that you have these significant local heating effects. So any changes that you're seeing in the catalyst or at the interface might not just be because of the reaction happening, but it might also be because of actual local heating effects by the incident source of energy. The other example here is this reaction here, where this was done at a synchrotron, but it's quite interesting to see. You have all of these interesting changes in your X-ray absorption spectra, which might be really tempting and might lead you to make certain conclusions. But what the authors in this study were able to show is that just looking at a UV vis of the catalyst, just in the presence of, or this copper complex in this case, in the presence of an X-ray without any reaction happening, already led to the decomposition and forming of these products that you possibly were related to some of these changes in the X-ray absorption spectra. So it's then important to have these control experiments in place to try and figure out that you're not really unintentionally doing something to your system and kind of leading to erroneous conclusions with that. So with that, I'd like to quickly summarize. There's a lot of different spectroscopies you can do. You can have X-rays which can ionize your sample. You can have all of these different UV-vis spectroscopies where you're looking at these DD electron transitions in our case. You can have infrared spectroscopies which look at molecular vibration. And it's really useful to then know what your open question is, and then know the knowledge and limitations of these different spectroscopies to then pick which one you might use for your system. And then in most cases, you often require using multiple different techniques and putting that together along with some theoretical insight in order to even try and get to what's happening at these really complex interfaces. And with that, Suren Scott is currently looking at ICSTAT, which is a place where you can start, or software where one can then start putting these different spectroscopies together and trying to look at similar measurement conditions across all of these different spectroscopies. And then finally, it's really important to know what you're measuring. Like last couple of examples, you really want to be careful about what you're measuring, have good control experiments in place, and really try and understand the technique that you're looking at to try and figure out what the data means. So with that, I'd like to conclude at least the first half of today's talk. And I'd like to thank all the people in the Durrant group and Ivan Stephens group, with whom I work quite regularly, as well as EL at MIT, which was where I did my PhD and where some of the examples that I've pulled out in this presentation were done. And then Sasha and Laya did all the nickel iron oxyhydroxide work. And then also Ben, Carlotta, and Soren are currently at Imperial and very much involved with some of this, the spectroscopies that we are looking at. So Ben is really good with all of the instrumentation. He's also looking at the nickel iron and the nickel oxyhydroxide catalyst. Carlotta has been looking at the iridium oxides, and then Soren, like I said a couple of slides before, is looking at this ICSTAT toolkit to try and combine these different spectroscopies together. So with that, I'd like to, I don't know if there's questions in the chat, or can have a break and then have Andreas do the second half.
Thank you, Reshma. That was really, really excellent. You can hear me, right?
Yeah.
Yeah, excellent. So we don't have any questions yet from the participants, but I strongly encourage everyone to send some questions. I have a question briefly. So I was interested to see what the slide you had about comparing using DFT to calculate the spectra. Mm-hmm. So for the UV-Vis, how often is that done, and how is there any cases where there's really good correlation between the experiments and the theory?
I haven't actually seen it done very often. They've just done it for iridium oxide, but they didn't have a lot of UV-Vis to support it. They just had more of the DFT. And then in the zinc cobalt case, they had some calculations, but it wasn't like computing the full spectra. But I think that's an interesting thing to do and to look at.
It would be really nice to have it on a very well-defined system, so that to see how well they compare with each other, because I'd imagine as well that and how it changes the function of potential.
Yeah.
Hi, Reshma. Here's Mongnan. I have posted a question in the chat, but I think I would rather ask here. So I think you, when you are mentioning the XAS measurement, like you mentioned something like a dip and pour technique, if I'm not very wrong, and it can be very largely affected by the environmental factors like the water, the moistures in the air or something like that. I think you mentioned that. And what could be done to minimize this effect?
Yeah, so people are trying, especially in terms of cell design, like trying to, so even the ones that I showed was like, really you need to have as thin a meniscus as possible, but then you want to still have a continuous meniscus, because if you don't, then you don't have ionic conductivity through the electrolyte, right? So it's, I guess the challenge there is mostly cell design and experimental design, and still kind of striking a balance between having significantly good electrochemistry, but then also being able to you can't have like several millimeters of electrolyte on the sample and then be doing those measurements, because then there's no way you can see anything from the surface. So I think it's still at a design of these beam lines and these cells, and trying to figure out what's the best way to do some of these measurements.
Okay, yeah, thank you.
Do we have any more questions? I have another question. Oh, sorry, we do have a question. Is there someone speaking?
Yeah, so sorry, yeah, I just jumped in. Hi, Reshma, very nice talk. I have a question regarding the in situ UV-Vis. So if the solvent or the electrolyte you are using that had the absorption in the same range, do you think that would be a large effect for doing the kinetic studies?
Yeah, that's a great question. I think if it's a constant background, I think it's something we can subtract out. But that would be another control experiment, I guess, to do to make sure that the background is not changing as a function of potential. But if it absorbs but always has a constant background, I think we should be okay.
Okay, thank you.
Hi, Reshma. This is Jesus. Thank you very much for the talk. It was really impressive. I had a question as well about the in situ UV-Vis measurements, because as I've seen, you use a transparent electrode that you put your material and then you analyze the changes. But when you measure OER, you may use an RDE or something else, which are different conditions. So can you claim that what you're seeing in a transparent electrode is the same that what it happens at the same potential in an RDE or a different electrode?
Yeah, that's also a great question. So we're currently also looking at not just doing it in transmission geometries, also being able to reflect off a substrate. So that would kind of remove this requirement of using a transparent electrode, and then one could have, for example, glassy carbon or gold or platinum substrates, and then try and get a bit closer to this kind of 3 electrode setup that we have, and also try and understand how substrates might influence kinetics, right? For a lot of reactions, we know, for example, the choice of substrate has a significant influence on the kinetics. So that would be, I guess, an interesting next step. And we're already looking, I think we already have this reflection geometry set up.
That sounds like a very interesting approach. Yeah, indeed. Thanks so much for the talk again.
Does anyone else have a question for Reshma? So I have a question. So obviously, when you have a lot of these different kinds of data, especially using electrochemistry and then maybe 2 or 3 different spectroscopies, I think there's a lot of talk about like using things like machine learning to combine it all together. Maybe you could use a kind of a platform like ICSTAT that CERN has been developing. How do you see that area moving forward? Or do you know of any good examples where people have done that?
I think it's a super exciting area. I think this is exactly what we need. And sometimes you have so much data and so much spectra that you're only looking for what your mind kind of tells you to look for. So you might be missing some really important parts of the spectrum, important parts of the data, which you weren't maybe looking for. So I think that's a really interesting area to look at. I know, for example, both with synchrotron and lab-based, there's a lot of push towards high throughput spectroscopies, and then trying to get thousands of materials really screened through and see how their properties change. And I think there was a couple, especially for oxygen evolution, which was done, I think, in Caltech, where they were looking at this high throughput approaches. But I don't know if any examples where they've really discovered. I think they're using it more as an approach now to benchmark what we already know, but looking at large number of materials, I don't know of an example where they might have found something new based off these approaches.